Hi, everyone. I am Miss Haley Kopczynski, and today I'm going to be presenting on signal transduction. More specifically, the topic at hand is going to be the cell cycle regulation in the cell. This is for Chemistry 530 Concepts of Biochemistry with Dr. Lee Ott. All right, a quick overview on the topics that I'll be covering throughout the presentation today is outlined here. Again, we are going to be talking about the cell. So I have a picture of the beautiful cell. We'll be looking at cyclin-dependent kinesis, checkpoints, cell size control, DNA damage response, monitoring DNA replication, meiotic spindle checkpoints, and diseases from dysfunctional checkpoints. So there are two classes of molecules that contribute to the division of cells. This is the cyclin-dependent kinesis and cyclins. Additionally, there are cell cycle checkpoints. These checkpoints aid in post-transitional modifications, structural biology, enzyme kinetics, and much more. In this presentation, as I stated, what we're going to be covering, cyclins will be first, and then cyclin-dependent kinesis. And then we'll get into talking about those different types of checkpoints that are there. And then lastly, covering what happens when those checkpoints don't work out. So diving right in, cyclins. Cyclins are cell cycle regulators and considered one of the most important. They are a group of related proteins. In humans, there are 4 different related protein groups. So we have our G1, our G1/S, our S, and our M cyclins. As you can tell by the names, each cyclin goes with the phase, transition, or set of phases it is named after. The cyclin aids in pushing the events in the cell forward that take place during that phase. An example would be that in the M phase, the M cyclin aids in what happens during the M phase, thus being the breakdown of that nuclear envelope and chromosome condension. So the picture to the right shows that there are different levels of each cyclin across the cell. When a specific phase is happening, that is when that cyclin that corresponds to it is going to be at its highest level. So for example, during the G1/S cyclin, you can see the peak level. You can see the peak level happens right in between the 2 phases, in between that G1, and you'll be able to see that. So if you look at the green line, the G1/S cyclin, that's where it's happening, right in between those 2 phases. And then as you can see on this red line, this G1 cyclin, that happens throughout the entire cell process because it's needed. So that one's kind of the exception there. Then we have our cyclin-dependent kinesis, and I just have a picture here because we can use that same example with our M cyclin phase, but I'll come back to that because I want to kind of explain the cyclin-dependent kinesis. So what it is, it's a family of enzymes that pair with the cyclins. So you can see the so it's called our CDKS. So you can see that is in this pink blob right here. Without a cyclin, which we already talked about, these are inactive, so it does not work. When you have the cyclin there, it's going to become activated, and then it's going to go to those activation sites. So one individual cyclin-dependent kinesis is inactive, but once bound with its cyclin, it becomes activated. At this stage, it becomes a functional enzyme, so it can be modified to go to those target proteins. So these cyclin-dependent kinesis, they're kinesises, which are enzymes that attach phosphate groups to specific target proteins. So that's why you can see that on the farthest right here. So when a target protein is attached to, that phosphate turns the protein, the phosphate turns off the protein, or it can turn it on and make it more or less activated. So they refer to this as like a light switch, if you will, because it's turning it on or off, so less or more. And when the cyclin-dependent kinesises attach to the cyclin, it activates it and it directs it again to those target proteins, which will then be in regards to whatever cell period it is in. So the example used previously was that cyclin during the M phase. It applies here too, because again, once those two combined, they're activated. So in this case, I have a G1/S cyclin, giving a different example instead of that M cyclin during the M phase. So checkpoints. Checkpoints are stages that the eukaryotic cell will go through, where it identifies both internal and external cues to tell whether or not to move forward with division. So there are 3 checkpoints that have significant importance, and that's going to be our G1 checkpoint, which is in the G1 or /S transition. So in between those two, because it's transitioning. And then we have our G2 checkpoint, which is at the G2 and M transition. And lastly, the spindle checkpoint at the transition from metaphase to anaphase. So at each checkpoint, they check for specific internal and external factors. So again, this picture to the right is just showing that where each of these checkpoints occur during that cell cycle. Right. Cell size is the first one that we're going to talk about as a checkpoint, or that happens within the checkpoints. So at the G1 checkpoint, this is where the cell is evaluated. At these checkpoints, which cell size has also been found in the G2 checkpoint too. So not to say that you can't find it in G2, because you can, it's in both. They look for genetic and biosynthetic material, which will be doubled before division. So we want to make sure that it's perfect, that there's no flaws in it before we start duplicating this and making more daughter cells. Cell size is important to maintain for the regulation of nutrients. And these nutrients are sent out, and the regulation organ size and function and multicellular organisms as well. So studies have found that the size of the new daughter cell after mitosis affects the cell cycle progression. So when there is a large daughter cell, the progression through G1 and G2, so the larger the cell, there's more progression. It will go faster. If the daughter cell is small, then it creates a delay when the cell is leaving that growth phase. So what scientists know about this checkpoint is due to the regulation of proteins that are involved in the G1 and G2 /M progression. So an example of this checkpoint is in fission yeast, because this is where it's heavily studied, is in looking at different yeast species. And the proteins that respond to the cell size and nutritional levels is, so these specific proteins are CDC25 and WEE1. And these 2 proteins are controlled by the CDC2 cyclin B complex. So cells control the cell size by protein translation or by monitoring the cell geometry. So an example of protein translation is also known as cell sizers. So this is our CLN3 and CDC25. So an example of how cells monitor cell geometry, though, is through a protein called POM1. And this is in fission yeast that they found it in. It's called S. pombre. So that's where it got that name from. The protein attaches to the tip of the cell, and it will stop the cell cycle progression through the regulation of its axes. So they have your CDR1, CDR2, WEE1, and CDC2 axis. So this complex is located in the central region known as the interphase node. When the cell length is longer, then the cell cycle can progress to M phase because that POM1 can no longer influence the complex. So this is again, this is something that's been researched in yeast. It's interesting because with just knowing how the cell monitors geometry, this could be a mechanism that is happening in other organisms, but we're not quite aware of that yet. So just some really an interesting study that's going on. But again, that other one is through protein translation through the cell sizers. And I have a picture of that G1 phase and where that is happening. All right, so DNA damage responses. So DNA damage responses are checked in both G1 and G2. So that's why you can see that on the last slide as well. So any DNA damage that is found will be stopped to allow for there to be a repair within the pathway. These are both intrinsic and extrinsic DNA damage. So intrinsic is anything inside of the body or inside the cell. So this could be attrition of telomeres, intermediates of metabolism, DNA replication errors. Any of those extrinsic, those are going to be things that are happening outside of the cell or outside of the body. And that could be sunlight, carcinogens, or ionizing radiation. So those are some examples there of that DNA damage response. And here in the picture, you can see if there's a DNA damage response, you're going to have, for example, P53, you're going to have an inhibitor. That inhibitor is going to say, hey, we got to stop this. We need to go back through and we need to fix the problem before it gets worse. So that's what's basically happening in short term. Monitoring DNA replication is another thing that happens during these cell cycles. So this is going to happen during that G2. So if the cell is considered voluble because DNA damage at this time can stop DNA replication from the following through. So we'll keep going. It will keep causing problems. So DNA replication happens at specific locations that are brought on by proteins. So the replication only happens once per cell cycle. And that's really important to know, as it only happens one time. This site is also known as replication origin. The replication origin firing occurs because of the two proteins that go through phosphorylation. And the proteins are CDT1 and CDC6. So these two proteins speed up the CDKs and the DBF4 dependent protein kinesis, CDC7. Therefore, the phosphorylation is needed to ignite replication. So it's just going to speed it up. It really just, it starts it, it's firing it up. Although it can go through degeneration of proteins, which stops the replication origin from refiring. So that's why it's a one and done deal. If the DNA damage cannot be fixed, the cell will undergo a programmed cell death, meaning that it's going to kill itself. It's no longer a viable cell. This way, the damaged DNA will not be passed down to the daughter cells. And this is actually a really important process that the body goes through because this mechanism prevents cancer. So it's a really important role there. Right, mitotic spindle checkpoint. This at the mitotic spindle checkpoint, the cell checks to see if the sister chromatids are attached to the spindle microtubules. The chromosomes must be securely attached to the spindle fibers from opposite poles. So again, you're one on top, one on bottom. Specifically, at least two before the separation of sister chromatids, which happens in anaphase. The way that the cell identifies if the chromosomes are attached is by actually looking at ones that are astray. So if they are out there free floating in the chromosome or in, I'm sorry, the cytoplasm, the cell's going to recognize, hey, this is how many we have in the cytoplasm floating around. So we must not have all of them attached. So the way, again, it will then wait, it will pause, it will stop it, kind of just like that inhibitor factor. It's just going to stop it and say, hey, you need the spindles need to attach to the chromosomes. And then it will recheck, and if everything's good, then it will keep going. All right, some defects. So there are times when checkpoints become dysfunctional, and what will happen is it's going to lead to cell death or cell reprogramming. Cell reprogramming, though, the downside is it may lead to cancers, and that's one of the big downfalls here if things aren't checked. And that's why we have checkpoints in our cells, to make sure that these things don't happen. So an example is the genetic defect P53. It's the most commonly found one in cancer. There are several cell fate decisions, which include CDKs controlled by P21. So this causes the G1 checkpoint to be lost. So if one checkpoint doesn't happen anymore, that means that there's errors that are going to be let through and things are going to happen. And then once it happens, you can't go back and reverse those. So just like talking about this, mitotic spindle checkpoint, once we get to, or once those spindle fibers are pulled apart, we can't put them back. So at these checkpoints, once something happens and gets passed down next, we can't go back and reverse that. So in summary of what I have presented to you today, cyclin and cyclin-dependent kinases bind and work together to activate each other. Checkpoints keep the cell in check to maintain healthy daughter cells. And cell death and cell reprogramming is what occurs from damaged DNA or an error occurs. So this can, again, those can result in cancers. And it's a wide array of cancers. Again, it depends on where you're at in the body and where those cells are and which ones are affected, what phase they're affected. So there's a lot of information out there. My references are here for you. So using some really great peer-reviewed journals here. Khan Academy is a really great resource if you're ever looking for a little bit less meat to the bone, so more of the bone in those. But these are some really great peer-reviewed journal articles that are very enlightening. And then as always, our Principles of Biochemistry textbook for you guys to reference back to. Thank you for tuning in.